that we welcome you to the second day of our National Symposium on Plant Biology in the Post-Genomic Era, Strategies for Crops and Mankind. After an eventful first day filled with insightful discussions and groundbreaking presentations, we are poised to delve deeper into the revolutionary advances in plant biology. This symposium serves as a beacon for pioneering research and collaborative efforts aimed at harnessing genomic innovations for the advancement of agriculture and environmental stewardship. Your presence and participation are indeed invaluable as we navigate these critical discussions and forge paths towards sustainable solutions for our crops, ecosystem, and well-being of mankind. May I now request Dr. Shobhana Chakraborty from Department of Physics, she's the head of the Department of Physics, to start today's first session on plant stress biology in the post-genomic landscape.
Thank you, Anindita. So, I am not sure if I fit into this, but yes, as a general student of science, I think I would be much honored to be a part of this symposium. So as Anindita already welcomed you all, so I would extend another, on behalf of Sister Nivedita University, not just the departments of biotechnology and microbiology, I would extend a warm welcome to all our esteemed delegates, my respected colleagues and our beloved students, to the second and final day of this symposium on, as already Anindita mentioned, plant stress biology in post-genomic landscape. So let me just quickly run you through the event schedule for today. That is, it's going to be held in 2 sessions. So session 1 is going to focus on plant stress biology in post-genomic landscape. It is going to be broken into 2 parts. So it's going to start or commence with a keynote lecture, followed by 5 invited lectures. And that's the pre-lunch session, and the post-lunch session will be session 2, which will again have 1 keynote lecture, followed by 2 invited lecture. And at the end of the day, you'll have the valedictory session and the closing ceremony to bring a closure to this wonderful symposium organized by the Departments of Biotechnology and Microbiology at Sister Nivedita University. So without wasting more time, let us just start the first technical session for the second day. So the first technical session is on plant stress biology in the post-genomic landscape. And it's with great honor I'd like to welcome, it's our real privilege and honor to welcome the chairs for this session. So this session will be chaired by Professor Somnath Bhattacharya, who's a professor in genetics and plant breeding in the Bidhan Chandra Krishi Vishwavidyalaya, West Bengal. And he has done his PhD in genetics and plant breeding as well. And this session will be co-chaired by Professor Shampa Dash, who is an INSA honorary scientist in the Division of Plant Biology at Bose Institute, Kolkata. So I would request the 2 chairs to please come on the dais. I would request Professor Bhattacharya and Professor Dash to kindly commence the technical session by introducing the renowned speakers for this session.
morning, very good morning to everybody. So today we are here for the SNU Bio-Talk 24, and yesterday it has been started. And today is the technical session 1, and theme is plant stress biology in the post-genomic landscape. So today's thrust area is plant stress biology in the post-genomic landscape. Actually, United Nations has set, you all know that sustainable developmental goals for the improvement and for the sustainable existence of the planet Earth. And for the sustainable improvement and sustainability of all life forms on Earth, we have to take enough protection for the plant kingdom. Because the plant kingdom is the integral part of this Earth, and without plant system, no life forms can exist in this planet Earth. In contrast, all the plant kingdom, all the members, rather, many of the plant members of this kingdom are being threatened by different biotic and abiotic stresses. So I congratulate organizers that the plant stress biology in the post-genomic, especially today's session is for biotic stress. So I congratulate the organizer for choosing the particular theme for plant stress biology, because plants are always threatened by different biotic and abiotic factors. So today's keynote speaker is Professor Alok Krishna Sinha. He is a renowned scientist of the country, and he is now staff scientist at the level of 7 at NIPG, National Institute of Plant Genome Research in New Delhi. And Alok Krishna Sinha is, okay, so it's very easy to read out. I don't read out because you all can see and read this. He is a man of map kinase. Really, he is a man of map kinase, and map kinase is a transcription factor protein, exists in all plants, and it has in all eukaryotes, and in plants it has multifaceted role, starting from plant development to response to plant stress, both abiotic and biotic. So he is, for quite number of years, he is looking for the identification, activity, and how this can be exploited, the map kinase can be exploited to defend plants from different kinds of stress. So his biosketch is in the screen, so it's very easy to read out his bio. I'm not going to. He is fellow of all national academies that I can declare here, and he also received different awards and honors in his career. In the early age, he got Alexander von Humboldt Fellowship. Actually, he received twice this Alexander von Humboldt Fellowship, and he got B.C. Dev Award and many, many, many awards. So please, Alok Sinha, for today's keynote speaker.
Hello, yeah. I think I'm audible, right? It's not a big code room. Thank you very much, Sampati, for this nice introduction and making my work a little bit easy by explaining what map kinase is. And thank you, co-chair. And yes, big thanks to Dhruvada and Arunda for bringing me or bringing us here at SNU in Kolkata. It was a pleasure to attend this conference and interacting with all the young faculties and students here. It was a very exhilarating day yesterday, which ended up with the cultural program from students of undergraduate, I guess, and fantastic talents and very impressive. It seems that the future of this university and the country is in safe hands. Fine. I'm very happy to be here and present a talk in this moot room. Why? Because my daughter recently graduated in the law from Symbiosis Law School, and she told me many stories about moot room. And I had no idea how this moot room looks like. So now I can go back and tell her that also I also defended something in a moot room. Okay. So I cannot say I, we, most of the speakers which come after me are from NIPGR, and this is how NIPGR looks like for a main building for students who wants to come to NIPGR for PhD. Fantastic campus, students in south of Delhi, in JNU, adjacent JNU campus, and this is sometime when the sun is setting in the west, and one of my colleagues took this nice picture of NIPGR main building. We have a campus where we have hostel for students and also residential area for staffs like scientists and technical officers. And we take students after MSc, we qualify some national exams, qualify our exams, join NIPGR, and fantastic institute with state-of-art research facilities for doing plant science research. Fine. So my topic, I think, today I'm talking about the regulation of cell cycle in rice by MAP kinase cascade, or I call it switches here. And again, for students, not for scientists sitting in the first, second row, that these plants, they seldom get such tender care. And they have to grow in a very harsh condition like this, where they have to face challenges from both atmosphere, biotic stress, abiotic stress, above ground stress, below ground stress, and this poor kitchen or source leaf has to do all this synthesis of food and translocate to the all sink tissues here to maintain the life cycle. And in this process, plants keep on signals from, variety of signals from everywhere. And this is what plant is doing since ages. But yeah, they are smarter, much smarter than what we are. They have a machinery established there by which they can sense this stimuli and send this message down to the nucleus for appropriate cellular rearrangements. And for doing this, this is one of this cascade, not only cascade, one of the cascade, which is called mitogen active protein kinase cascade or MAP kinase cascade, which is a versatile cascade. So what happens here, again, this is any stimuli which is perceived by the receptor activates the first member of this relay cascade. First member, which is mitogen protein kinase, triple kinase. This triple kinase, upon activation and phosphorylation, activates MAP double kinase, which then activates MAP kinase, the last component of this cascade. So once this MAP kinase is activated, it interacts with various cytosolic protein and also nuclear protein, mostly transcription factor, and regulate almost everything what is there in your plant system. You call it root development, apoptosis, small level, biotic stress, abiotic stress, and several other things. So whenever I saw this slide, it reminds me of a very nice advertisement in TV of Mr. Amitabh Bachchan, where he comes with a just dial, that if you have a just dial, why do you need other apps? So when you have MAP, you don't need anything else. It takes care of entire plant development. Yeah. So coming, and if you talk about rice, I'm just confining to rice today because we work in my lab in rice and Arabidopsis. Today my talk will be confined to only rice. In rice, it is a multigene family. MAP triple kinase is 75 members, MAP double kinase is 8 members, and MAP kinase is 15 members. And these are divided into different classes. And some of them are very well characterized, but still much is still to be deciphered. Fine. And then if you come to my lab, what we are doing for last maybe 2 decades in NIPGR, my entire work is confined on this MAP kinase cascade, and we look at different aspects of this MAP kinase. Maybe it's submergence, hormone signaling, biotic stress, abiotic stress, light signaling, also in microRNA biogenesis, and cell cycle, which is recently we call it in plant biology. We started in 5-6 years back, but it takes time to get your transgenics and everything mature. So I call it recently, and we have certain publications on this. Submergence, we have a very interesting story where sub1A1 is controlled by MAP kinase and helps in submergence tolerance. Then in light signaling, we were first with Sudeep Chattopadhyay, and Vishmita was the one, his husband is sitting here. She helped in identifying first time the activation of MAP kinase by light, which interacts with a MYC2 transcription factor and negatively regulate entire blue light morphogenesis. And then we have certain other papers coming in the same directions after that. Fine. So today I'll confine to this cell cycle only and MAP kinase. And I'll try, my talk will have a lot of data for those who are actively in research will slightly appreciate that, but those who are not in this active research, just follow what I want to say. So in the next 20 minutes or so, I'll try to establish what I want to convey. So what is happening here in cell cycle and MAP kinase? If you look at the literature available about involvement of MAP kinase regulating the specific component of cell cycle in animals, there are many reports where you have MAP kinase interacting with P27, KIP is another one, then E2F1 by another MAP kinase, so V1 inhibitor of cell cycle, and also from yeast. But when we come to plant cell cycle and MAP kinase, the information is very scanty. And there is no information of a direct regulation of a cell cycle component by a MAP kinase. You have some indirect, like MAP kinase 4 is required for cytokinases, and this module of YDA is a MAP triple kinase, MKK4 and MPK3. They regulate in root apical meristem, but not actually looking at the particular component of cell cycle which is being controlled by MAP kinase. Now, if you look at this, I think most of you are aware of this, MG1S G2 cell cycle, and you have different proteins here involved which controls the movement of cell cycle from G1 to S, S to G to G2 to M phase, and these are cyclin dependent kinase which are 4 members in plants, CDK, B, C, and D, which combine with cyclins there and perform this downstream function. They have certain transcription factors like E2F, MIC here, BIB here, which regulates certain genes of this cell cycle component and changing this G1 to S phase and all. And then there are some inhibitors. The KRP is KIP-like related proteins in plants. In animals, this is KIP proteins, V1 and SMR1, which are inhibitors or regulating cell cycles. This is what is known in plant. If you come to animal, there are fewer genes, but almost similar genes doing the same role. Now, if you look at the information available about MAP kinase regulating these components, there are some reports like ERK1 regulating cyclin D, ERK1 there 2 and P38, interacting with V1, KIP, RBR, E2F and all. So this is there. So much is known about animal MAP kinase regulating cell cycle component, but nothing in plant. Now, what happened is that there are so many noise about, and papers keeps on coming about this cell cycle in animal and MAP kinase. But if you come to plants, there is a quite good silence. So this was our question when we started about to investigate and moving into from whatever we are doing in the lab to cell cycle. And it started with one of my students who was looking into effect of UVB rays on rice seedlings after giving UV rays for 1 hour, 16 hours, doing a global transcript profiling. And as well, you find certain genes going up, certain genes going down. What caught our attention is that there are certain genes involved in cell cycle components, they were co-regulated with MAP kinases. This was CDKA and some of this inhibitor V1 and SMR1. And also it induced serotonin, which is a different story, I'm not touching that. So it was like MAP kinase was found to be negatively regulating UV-based stress. Now, we found that this CDKA showed a upshift in a SDS page when we do with a domain antibody, that is shifted up. So we thought that this might be a target of MAP kinase. So what we do first is that we do a protein-protein interaction by yeast-2 hybrid or BIFC or pull-down assay, and we found that no, this CDK is not interactive with MAP kinase. I'm just showing a yeast-2 experiment here. We also did BIFC and tried to dig it out whether we are doing some mistake or not, but there's no interaction with this. Then we moved to the other 2 inhibitors, V1 and SMR1. This is a pull-down assay which showed that by just SMR1 having a GST tag, and then we pulled down and we found that this is attached with MPK3 and MPK6 in a pull-down assay, which was there also in yeast-2 hybrid and also in a BIFC in planta assay. And then we confirmed it by cloning them, expressing them in a bacterial system and doing an in vitro phosphorylation. It's an assay for identifying whether this particular protein is a substrate of kinase or not, where we make this protein in bacterial system, purify it, mix with the kinase in an ambient tight buffer, and then you, in the presence of radioactive ATP, or there are other techniques also, you can find the transfer of phosphate from ATP to the substrate, which shows a signal like this. So SMR1 gets phosphorylated. That indicated that yes, SMR1 is a phosphotarget of MAP kinase. And also similarly, we also identified that V1 is also a phosphatidylate of this. This was not actually, we were trying to identify the function, what happens after phosphorylation. So we reported this entire finding recently in PPB, and Gopal Banerjee, your student, was, I'll come to the next story. We took it further. We also found that this is the first part, that we identified MAP kinase phosphorylating V1 and SMR1, two components of cell cycle. Next, in another experiment, I'm just cutting down story short, by feeding this rice seedling with inhibitor of MAP kinase, we found that out of this 4 member of E2F, which is a transcription factor, only E2F2 is showing the same upregulations as MPK. DP1 is another dimerizing protein which dimerizes with E2F for its function. Then it binds to the promoter of different genes of cell cycle for its transcription. So these two, DP1, behave similarly like MPK4 and MPK6, but MPK3 was slightly different, showing up to the same time point. So again, to the students, this MPK3, MPK4, MPK6 will keep coming. These are different MAP kinases. MPK3 and 6 belongs to same clade, clade A, and MPK4 belongs to clade B. Sometimes they work synergistically, sometimes they work independently. And it's very difficult or interesting to identify when this MAP kinase is phosphorylating which protein and what for. Okay. So we found that there's some co-regulation, and then we look for the interactions. First thing, that these two proteins are interacting or not. Yes, they are interacting in this system, not only with all this MPK3, MPK6, MPK4, all this E2F is interacting. In planta also by BFC, and also this MAP kinase are interacting with the DP1 protein. So these two are being regulated by MAP kinase, maybe, and then we look for the phosphorylation. Now you see here, this is, I'm just making the slides very busy because I have, as I told, that there are a lot of data coming up, and try to understand that we are trying to establish that this particular protein is indeed a target of MAP kinase. Now, if you look at this, that this E2F2, wherever you add E2F2, you will get a signal here. This signal is only of MAP kinase because MAP kinase shows autophosphorylation. And to confirm that MAP kinase is the active one, we also do a control experiment by using its known substrate called myelin-basic protein. Well, fine, this is here, downstream. So we found that E2F2 is getting followed by MPK3, this case, and MPK6 here. Plus sign indicates presence of protein, minus signs absence of protein. And also by MPK4. Now, again, we identified, looking at the sequence, what are the possible sites where MAP kinase can phosphorylate. So this can be done by looking, because MAP kinase phosphorylate mostly in a serine or threonine residue, which is followed by proline. But then many things are also required. So we identified there are many SPTP motif in E2F2, out of which 3 of them were very conserved. So this serine 40, threonine 44, and 3.84 were highly conserved. So we mutated this into alanine. Now, if you change this serine into alanine, it cannot be phosphorylated. We call it phosphodead form. And when we use this phosphodead form, the phosphorylation of E2F2, this upper band, goes off in case of MPK3, MPK6, and MPK4, but not in MPK3, indicating that MPK3 might be phosphorylated at some other place. And this was also confirmed by a phosphate gel. Phosphate gel is another assay, which is different than SDS page, which can separate this phosphorylated protein from non-phosphorylated protein. And again, it's got the same result whenever we have MPK3 and MPK4 and MPK6 and use a phosphodead E2F2, this phosphorylation upper band goes off, indicating that this is being phosphorylated. Now, to confirm that this is actually involved in DNA proliferation, we do an EDU experiment by adding EDU, which is the thymidine analog, and gets incorporated in DNA when it multiplies. And this can be seen in a microscope after adding with a Alexa-Flor dye. So when we add this vector control, this is when we transiently overexpress with E2F2 and E2F2 phosphodead form. Whenever we use phosphodead form, the DNA proliferation is more. In case of only expressing MAP kinase, it goes down. In case of expressing MAP kinase with E2F2, it again goes down. But whenever we express this in the presence of dead form of E2F2, the DNA proliferation is more, indicating that MAP kinase phosphorylation negatively regulating DNA proliferation. And then we found that this E2F2 not only regulating cell cycle genes is also regulate MPK4 by binding to the promoter by Gus activity. And this also we just reported that now we have 3 proteins. I talked about V1, SMR1, and now E2F2 as a target of MAP kinase. So my dear friend Devasis always says that MAP kinase phosphorate anything. It's not like that. Devasis not always, because out of these 4 members, E2F2, 1, 2, 3, and 4, it only targeted E2F2. Fine, this is done by Dhanraj, and yes, we also reported last year. I wanted to have that, yes, we have a report of MAP kinase phosphorylating cell cycle components. And then we moved ahead, and in our experiment where we, seedling experiment where we treat them with the inhibitors of MAP kinase and cell cycle. This is MAP kinase inhibitor, cell cycle inhibitor. We found certain of these genes getting upregulated very strongly, CDKD and KRP3. CDKD is one cyclin-dependent kinase, and KRP3 is the KIP-related protein, which is an inhibitor of cell cycle. Now, we also checked what happens to the MAP kinase when we do the same, and again we found with this cell cycle only MTK3 is coming up, but among this MAP kinases, all 3 MAP kinases, 3, 4, 6, are transcriptally upregulated. Now we checked whether this has an interaction with KRP3 or not, and interestingly, out of 8 members of KRPs, only MPK3 interacted with KRP3. Which is very, very specific. Only MPK3 and KRP3 are interacting, not MPK4 and 6 with this KRP3. And then again, we proved it the same way and looked at the phosphorylation. We found that KRP3 is phosphorylated whenever we have MPK3 here, and also here. I mean, we are using here to activate MAP kinase by using MAP double kinase continually active. This was asked by one of the reviewer. Not really required, but yes, we did that. So KRP3 is phosphorylated by MPK3. Fine. Then we identified that this KRP3 has 4 putative phosphosite of MAP kinases at serine 17, 82, 1112, and 168, all serine. So we've generated several mutants of this KRP3 by doing a side active mutagenesis where we kept one active and made other 3 phospho-dead. Like you see the black one, they are all phospho-active, there's wild type, and other were made change into alanine. So if we get a signal from, phosphorylation signal from here, we can understand which one is getting phosphorylated. And then the final one, we made all of them kinase dead. And then we did the assay. You can see that wherever we have changed serine into serine 17 and 82, the phosphorylation is lost here. So you can see a signal here, because here either S17 or S82 is present. Here 82 is present, 17 is present. Whenever we have both of them 17 and 82 mutated, there is no signal. So now confirming that this is the MPK3 is phosphorylating at S17 and S for 82. Fine. So this is again good that MAP kinase is phosphorylating one other member of inhibitor of cell cycle component, but what for? So what happens for post-phosphorylation? It may change its localization, or it may change its interacting partner, or it may affect its stability. So we checked what happens to its localization by making this KRP3 phospho-dead, where these 2 amino acids will change into alanine and also phosphomimic. In case of phosphomimic, what we do is that we change this serine or threonine residue into aspartic or glutamic acid, which mimics the phosphorylation state, and you don't need an upstream kinase to activate it. So it remains in activated form. So we made this 2 construct of KRP3 phospho-dead. A is phospho-dead, E is glutamic acid, which is phosphomimic. And there was no change in its form, either in phospho-dead form or phosphomimic form, in its localization. Fine, it's not changing its localization. We looked for its interaction with other protein like CDK and cyclin D2, but there was no change in the interaction also with this 2, so phosphorylation is not affecting that. So what happens to the stability? Then we found that yes, stability is, when it is phosphorylated, it is more stable. This is a assay where you take the protein and mix it with the plant protein extract, and for the certain time period, and then do a Western to identify the amount of protein present. So the wild type, you can see at 180 minutes, everything is gone. In phospho-dead form, it is reduced substantially, but in phosphomimic form, it is still there after 180. So it somehow plays a role in making this protein stable and having more activity. Now, fine. So this is all we had. So we wanted to find out further information about what happens to this phospho. So what we did is that we generated CRISPR mutant of this KRP3 also of MPK3, which was interacting partner, and a double mutant of MPK3 and CRISPR, KRP3 by CRISPR-Cas. So we heard about this genome editing tool yesterday. I mean, it's just a, we call it genome editing tool. It is a tool. The way we make transgenic, similar way we make CRISPR-Cas9 editing lines, and it is important to know what is your question and why do you want to make it. And making it is not difficult at all. And in parallel, we also overexpress KRP3, KRP3 dead form, and KRP3 mimic form in plants. So we had 6 lines of transgenics, and then tried to see what happens to the plant growth and development when we are altering this KRPs and MPK3. We thought that this is a cell cycle inhibitor. If we knock out KRP3, it will help in the more growth of the plant. The more cell cycle division will be there. But to our surprise, even both KRP3 knockout and KRP3 overexpression showed similar type of phenotype. Your plant height went down in both the cases. Tiller number was almost going down compared to the wild type. Also, this internode length. But the difference came out when we saw the leaf length. The leaf length, when we have a KRP3 overexpressed, which is the inhibitor, was substantially reduced compared to the KRP3 knockout. Again, we did this self-proliferation assay. Again, we found that both KRP3 knockout and overexpression line, the cell proliferation goes down. So this was very difficult for us to explain what is happening here. Now, we compared this with KRP3, MPK3 mutant and double mutant of MPK3 and KRP3, and all of them behaved similarly. All the phenotypes were almost similar here, killer number, plant height, some different internode, indicating that both of them are playing a role in the same pathway. And even this, if you look at the maintaining pollen size, you have aberration in pollen size also in all these mutants, MPK3, KRP3, and double mutants. And also its seed size and substantially reduced compared to wild type. So both of them are playing similar role. So not much information was could be achieved from this mutants and hard work of 2-3 years during COVID time. Then we compared what happens to this when we overexpress KRP3 dead form and KRP3 mimic form. In this case, we found that whenever we have this KRP3 phosphomimic form, the plant height was substantially reduced. So very pronounced effect of if you keep this KRP3 phosphomimic, which makes it more stable, then your plant height goes down. And the reduction in leaf size was also very, very strong. Even the seed size. So the phosphorylation of KRP3 is required to maintain stability, which then inhibits it adversely for the plant growth and development. And this was done by Gopal Banerjee, and we are still working on this and still unpublished. And they're not talking about any flowchart here. Okay, so we did something extra on this KRP3, made a lot of transgenic and got information about that. So now this is the fourth protein. Now I come to the last part of my talk in the next 10 minutes. This is now, we got, if you remember, there's a upregulation of CDKD along with KRP3. So CDKD is another member which showed very similar expression when we feed this rice seedling with cell cycle inhibitors. So all these 3 and 3, 4 and 6 coming up, not 7 and 14, another member of MAP kinase which belongs to group C, but CDKD is showing a similar pattern as MPK3 and 6. And we can see the activation of MAP kinase by adding an inhibitor, and this activity was identified which MAP kinase by using specific antibody. It was MPK3, 4 and 6, but not 7. So again, by transcript and by looking at the antibody, we can confirm that MPK3, 4 and 6 getting activated. Similarly with the upregulation of this CDKD. Now we looked into the interactions. I will not go again in the detail. So we found the interaction in IS2, in pull down assay with CDKD with all these 3 MAP kinase, also in BIFC assays. And when we check this phosphorylation, interestingly, CDKD itself is a kinase. So you get an autophosphorylation of CDKD. When we added this with, this is MPK3 alone, and we added both this together, we see another very strong band signal, more than the wild type, indicating that it's getting phosphorylated, but you cannot be very sure because CDKD has this autophosphorylation. Then we checked it with the phosphoserine and phosphothreonine antibody because it had some serine and threonine sites, and we could see a stronger signal when we use phosphoserine or phosphothreonine antibody, confirming that CDKD is getting phosphorylated by MAP kinase. But to have this, to get this real information, what we did is that we took this this CDKD, and by our in silico analysis, we again found that there are 4 putative sites, 3 of them serine and 1 of them threonine, which could be a putative target of MAP kinase. First, what we did is we made this CDKD autokinase dead. For this, we changed this lysine residue into arginine 48 so that there will be no autophosphorylation. Now, when we use this protein as a substrate and we get a signal, we can say for sure that this is because of MAP kinase. And then we mutated all 4, making kinase dead, and again serially keeping 1 active, making other 3 dead, and then identifying which sites. So a lot of mutations were done here, systematically cloned, expressed, many times had a problem in getting the assays, and this radioactive assay is not that simple, and the student was fantastic at keep doing this assays many times, because we always believe that you have to get that signal which can be published, not any signal which only indicates that it is phosphorylated. Fine, so these are pretty good signals, and again, this is now CDKD kinase dead form, which is not showing any autophosphorylation, is getting phosphorylated MAP kinase. So giving a good evidence that yes, it is indeed a target of MAP kinase. When we change all of this 4 sites into kinase dead, there is no phosphorylation. Again indicating that one of these sites could be a site for MAP kinase phosphorylation. Similarly, we did it for MPK3, 6, and 4. I won't go in detail all this because a lot of data now again interacting is 2 and yeah. All right. Yeah, fine, sorry. So what happens to this if we change this into phosphomimic and phosphodead form? Again, we found that there was no change in its localization, not even its interaction with CDKD diamine with cyclin H protein. Then it phosphorylated CDK and B for further progression in cell cycle. There was no change even in this interaction of CDKD phosphodead or phosphomimic with cyclin H. But we know from Arabidopsis that CDKDs also phosphorylate C-terminal domain of RNA polymerase II. And this is required for processing of microRNA. We checked what happens to this. Is it true in rice also? That the CDKD can react with RNA polymerase II, and we could see that this is CDKD with RNA polymerase C-terminal domain. There is an interaction here, and also with cyclin H. So this was known in Arabidopsis. We just tested whether this is true in rice or not. It was also interacting also in rice. And then we checked what happens to the phosphorylation of this RNA polymerase C-terminal domain by CDKD. You see that if you have CDKD here in RNA polymerase II, it gets phosphorylated. All right. But then after identifying its site, I missed that it has a serine 168 and 235 as a MAP kinase target. So MAP kinase phosphorylate CDKD at serine 168 and threonine 235. This was done by, again, this SDM mutagenesis and all. And when we use this kinase dead form of CDKD, it fails to phosphorylate RNA pol 2. So indicating that phosphorylation of CDKD by MAP kinase is required for the phosphorylation of RNA pol 2. And when we add cyclin with the CDKD, the activity of CDKD goes up. And you see here, when we have cyclin included here, this phosphorylation goes up. This is very, very strong phosphorylation of RNA pol 2. But even in the presence of cyclin H, these kinase dead forms are not phosphorylated by CDKD. So this phosphorylation of CDKD by MAP kinase is required for the phosphorylation of RNA pol 2. And now, after having all this information, we again generated several lines of transgenics where we expressed the kinase dead form of CDKD overexpression line. This is D is we change into aspartic acid. This is phosphomimic form. A is alanine. This is phosphodead form, and this is kinase dead form of CDKD. And analyze what happens to them during their growth and development. And wherever you see that, wherever we have CDKD overexpressed or CDKD phosphomimic form overexpressed, you have a positive effect on the plant growth and development. When we have a dead form and CDKD dead form, there is no difference with the wild type. It's not negative, but no difference. But in the overexpression line and phosphomimic form, it is always more in whatever we say, plant height, plant panicle number, seed numbers, tiller number. but not on seed size. So this CDKD, which is phosphorylated by MAP kinase, helps in plant growth, but not in seed development. All right. Now, what happens here is CDKD phosphorylate RNA pol 2, which helps in the 5' RNA capping of this, and this precursor of microRNA is processed and mature RNA is formed. So if you interfere with this, you will have less of mature RNA, mature microRNA, and more of precursor microRNA. And this is exactly what we found, that in this transgenic line, all this microRNA which are known also were significantly reduced in case of phosphomimic form. And also in this kinase dead form. So what we identified is that MAP kinase phosphorylate CDKD in these two residues, which is not affecting in its localization and all, but it phosphorylate helps in phosphorylation of RNA polymerase II and processing of microRNA. And this is all, so what finally was done by Dhanraj. So this two part, Dhanraj and Gopal did all this work. And what we come finally to summarize my talk, that when we started understanding about this cell cycle component by MAP kinase, there was nothing known, but now we have MAP kinase phosphorylating V1, also SMR1 to E2F2 and DP, and then to CDKD and also cyclin H. So we have at least stamp at 6 different proteins of cell cycle component by MAP kinases. So this is what we started, and now we are about to, this is also two of this last part is still to be published. And yeah, thank you very much for your attention. And before I complete, I'd like to acknowledge my students, Gopal Banerjee and Dhanraj, and these other people who are working, helping in making transgenics, and also some of them are working in different aspects of Arabidopsis MAP kinases, which I'll get thanks. And thanks to the funding from DBT, SERB, and from our institute. And thank you all very much for present hearing. We will be happy to give any clarification or answer if there's any doubt.
Thank you, Professor Srina. It is a really nice talk, particularly the new dimension of that MAP kinase in cell cycle regulation and other things. So now it is open for discussion or any queries or any comments on his great lecture. On the student side, if there is any comments or any queries, please. He is the authority of, I mean, not only nationally, internationally, he is the authority for MAP kinase, you can say.
RNA cannot be. Yeah, Jiten.
RNA pol 2 C terminal end, it's phosphorylated by so many kinases. Now you have found one MAP kinase. In the cascade, there are so many others. Do you see the sequential kinase by others, MAP KK, MAP KKK, MAP K4 and all? You tested only one MAP kinase. What I'm saying that in the context of regulating cell cycle genes, do you see sequential phosphorylation, dephosphorylation of RNA pol 2 CTD by other MAP kinase cascade?
Yeah. See, just to make it better, we are not, I'm not saying that MAP kinase phosphorylating RNA pol 2. MAP kinase phosphorylating CDKD. This phospho-CDKD, MAP kinase phospho-CDKD is phosphorylating RNA pol 2.
Okay, it's phosphorylated by CKD.
CDKD. CDKD. So your question is valid, because I always feel that we open only one window and see what can be seen from there, and say that this is working in the system. But a system is not working in isolation. So the experiments what we have performed here indicates that unless and until this CDKD gets phosphorylated by MAP kinase 3, 4, and 6, it is unable to phosphorylate RNA pol 2. But the possibility of involvement of other kinase cannot be ruled out. And here we are not trying to look at upstream kinase at all. This is too much of work to figure out which MAP double kinase, which MAP triple kinase. So this is another work which is open for the students to come in subsequently.
This is one of the most preferred phosphorylation target.
Yeah.
So, yeah.
So along the MAP kinase cascade, so the phosphorylation comes from top to bottom. So is there any evidence that the last one, the third enzyme, also reversely phosphorylates the second one and second one the first? Is there any report? Did you get my question?
Did I told you something like that?
No, it's a generalized question.
But then it's good.
Yeah.
We have found it.
Okay.
And I'm confused about this. So I found in one case, in submergence case, MAP kinase 3 is phosphorylating MAP double kinase.
Yeah.
So this happened many times. I was trying to look whether double kinase phosphorylating MAP single kinase, but it happened other way. Then we mutated this MAP double kinase and could prove that it's actually phosphorylated by MAP kinase. We also have evidence of, in another rank, we are also working on involvement of MAP kinase in photosynthesis, where we identified a MAP triple kinase, which is a target of MAP kinase. Now, what it does, we don't know. This is what we're trying to do.
But it happens.
But this happens. This is happening in my lab. I have not seen any report so far.
Okay.
This is why I asked whether you. Huh? The canonical cascade is from MAP triple kinase to this.
That's okay. Canonical is always means when you discovered it and how you studied it. But it cannot be confusing. All the kinases, or if you treat them as an individual protein, they can be target of any kinase in that cascade.
Yeah, agree, completely agree. But when you look at the literature, all the informations are like that. There is no such report so far where you have a inverse phosphorylation and leading to a biological response. We have found it. When I got it, I was confused because I tried many times, many different cascades. I never observed this reverse phosphorylation. Now we have information about this, what happens after reverse phosphorylation also. So we have made this transiting, and in case of this MAP triple kinase, upon phosphorylation it goes somewhere else. When it's phosphorylated by the receptor kinase, it goes to the cascade. So, yeah, somebody.
For your complementation, as in the pull down, in complementation you added MPK3 from OS MPK3, but MPK6 and MPK4 you add Arabidopsis.
No, not Arabidopsis.
80 MPK6 or 80 MPK4 you add?
No, okay, fine. So it may be one of the Western. We have antibodies of rice MAP kinase of MPK3 working very nicely. But the antibody of MPK4 and 6, what we generated for rice, is not showing very nice positivity. It shows a lot of band. But the commercially available antibody of Arabidopsis of MPK3, 4, and 6 can cross-react with rice MAP kinase. So that must be a Western blot where we might have used 80 MPK3 and 90 MPK6 antibody only, which cross-react, which we have shown in our lab by doing an in vitro expression of this kinase in bacteria. Yeah. Thank you. Any question for students? Even what is MAP kinase is a question.
I think if you can explain the difference in the results between the phosphodate versus a phosphomimic.
All right, once again.
So what actually it signifies?
See, since I got this opportunity, I'll tell something more. That plant is a wonderful system. It's very mysterious. And the more we work, the more questions we raise. And I encourage all students to also to choose the plant as a research career. Not because I am working in that, because if you can transform rice variety type I1, you won't be able to transform rice variety Swarna. So every variety is different. It's not that you transfer my cell line, it works. Now, in my case, what we are trying to do, we're trying to look at the phosphorylation target of MAP kinase. MAP kinase is a kinase. Its job is to phosphorylate. What it does, it transfers phosphate from one protein to another protein. Now, there are certain amino acids which are labile for phosphorylation, commonly serine, threonine, and tyrosine, because they have a OH group which can be easily replaced by phosphate group. Now, this serine residue or threonine residue, which is a target of kinesing or phosphorylation, if we change this serine into alanine, it becomes inert. Now, this alanine cannot be phosphorylated, so it becomes kinase dead. Okay. If we change this serine or threonine residue by aromatic acid, either with D or E, that is aspartic acid or glutamic acid, it mimics the phosphorylation status. So you don't need upstream kinase to phosphorylate and activate this protein. If you change the serine threonine into aspartic acid, which is D, glutamic acid, which is E symbol, then it behaves like a phosphorylated protein and can activate downstream processes. Yeah, because of negative charge. Yeah. So this is easy way of looking at the function, what happens post-phosphorylation. All right, and this involves a lot of work. I mean, this is a lot of cloning, a lot of expression. Okay, thank you very much. I hope I'm not very late.
So thank you, Professor Sinha. So before we move on to the next session, that is the invited talk, which we'll have soon, I would request our Honorable Vice Chancellor, Sir, Professor Dubajyoti Chattopadhyay, to kindly felicitate our keynote speaker. Along with the two chairs, Professor Bhattacharya and Professor Dash.
So we will not break right now, just another lecture will be delivered by Dr. Devasis. Okay, just finish this talk, then when we will break, then we will just make the podium ready. It's just 20 minutes. Okay. So I request Dr. Devasis Chattopadhyay for the next talk. He has also very illustrious, career in the plant science, and he has very well-known face for all, in our national level, those who works on plant, he's a very well-known face, and he also got all sorts of, fellow for in almost all academies, and we are really pleased that Devasis and almost all the leading plant scientists has invited, and they have agreed to join in this very important seminar. So Dr. Devasis, you know that he is a now staff scientist at National Institute of Plant Genome Research. He is also staff scientist 7, and he has a lot of awards and honors. And particularly, he is very much well known for his abiotic stress tolerance and how the different genes and all other genes interact with each other. And I also know very, closely for his arsenic related work. He has a lot of arsenic related work also in earlier days. Okay. Devasis at least for lectures.
Good morning, everybody, and Sampadi and Samradha, and thanks the organizers for inviting me. And I did PhD with your Vice Chancellor, and I am the only black sheep from his lab who converted to plant scientist. So, so my apology for that. So this is the plant biology in the post-genomic era. Post-genomic era, that means now we have the genome sequence and the gene sequence of almost all the common crops which we use. So we are trying to translate some of our results from the model plant, which is Arabidopsis, to the crop plant, and then we are facing the problem. So just today I will show you that what is the difference between, at least in one aspect, the lignin deposition in the root, what is the difference between the working with the model plant and the crop plant, which is chickpea, which is the chana or chola, whatever you know. So I work with both the systems. So not only the species difference, you see the growth condition difference. When we work with the model plant, we grow them in the petri dish with the synthetic media, and the roots are growing under light, which generally does not happen, right? Generally the root grows under the soil. But when we grow the crops, this is the system, right? Not only the species difference, so this system also matters. So now I think most of you may not be the plant biologists, so I was told that I should keep my talk less technical. So I kept the very easy slides, and generally whenever you apply any stress, any drought or salt or this kind of stress, then generally you will see the lignin deposition in the vasculature, in the xylem mostly, in the root, also in the shoot, but today my talk will be only on the root. So this one is okay, yeah. So you can see the lignin deposition in the vasculature, and this is the with the PEG which mimics the drought, and there is a lignin deposition and lignin deposited inside the primary cell wall, that is called the secondary cell wall. So with towards the cytoplasmic side, so lignin is deposited. Now what is the advantage of lignin deposition? Because the water pressure in the given conduit is inversely proportional to the fourth floor. It is like a tube well. So if you reduce the radius, so your water pressure will increase because the plant needs more water during stress, right? And the second reason is the lignin is hydrophobic. It is made up of the aromatic amino acid. So it is hydrophobic. So during the water transport, so there will be less leakage of water. So these are the main advantage of having lignin deposition in the vasculature. So as I said, it is from the aromatic amino acid. It is, I think, it's biosynthesized from the phenylalanine, and there are a number of steps. And the last step is the catalyzed by these two family of enzymes. One is the peroxidase, which utilizes the H2O2, and the lacase, which is the molecular oxygen. So my talk will be on the lacase today. And this is the reversible reaction. And from monolignol, it will be monolignol radical, and then it will be polymerized with the cellulose and hemicellulose of the cell wall, primary cell wall. So this, as I said, that this is the reversible reaction. In most of the fungus, you will see the lacase that degrade the lignin, because that's how they infect the plant stem. But in case of plant, it was found there is a prolignin. That means if you knock out, for example, Arabidopsis has 17 lacases. If you knock out LAC4, LAC11, LAC17 triple mutant, then you see no lignin deposition in the xylem, and the plant do not grow more. There is a xylem collapse, there is a vasculature collapse, and the plant die in the early stage. Okay. And the double mutant or the single mutant do not have any phenotype. That means they have a redundant function. So that means whenever you have the triple mutant, then only you see there is a total no lignin deposition in the root. So now, let's start the story from the Arabidopsis, the model system, what happened in my lab. So I treated with the mannitol, which is the osmotic stress, and this is the normal growth of the plant, but whenever we treat with the mannitol, the plant root growth stops, everything on the root today, okay. So the root growth stops, and we see there is a lignin deposition in the xylems, okay. And this is only in the local area, but whenever we chemically estimated the lignin content of the whole root, so this is also, you see the increase in the lignin deposition. And this is the closer view. You can always see, sometimes you can see the double xylems also, okay. Now we tested the expression of all the 17 lacases, and surprisingly found that in the root, only these two lacases, LAC2 and LAC4 is down regulated, not up regulated. There are other lacases which are up regulated or remains the same, but this LAC2 and LAC4, they are down regulated only in the root and only in these mannitol stresses. Okay. So we first brought this LAC2 knockout mutant. I'm sorry because this has moved. These two are the LAC2 mutant. And this is constitutively, you can see that the shorter root, and but lacase should be a prolignin, right? If you knock out, so there should be no lignin, but it opposite the if you knock out the LAC2, you see the more lignin in the root. And when we complemented with the lacase2 cDNA in the knockout, either on with the 35S promoter or its own native promoter, again the root rescues its length. So normally in constitutively, this LAC2 knockout mutant have more lignin. So this is the fluorescence stain, and as I said, it is made up of the aromatic amino acid. You do not need any stain. If you expose it to UV, you can easily see this lignin without any staining. So that means the LAC2 is a negative regulator of the lignin deposition. And what is the mechanism? We still do not know what is the mechanism. How out of all lacases, LAC2 is the negative regulator. Other lacases are positive regulator of the lignin deposition. We are still working on it. Why or how this lacase 2 is the anti-lignin deposition. And if you search in the literature, you will see there is a microRNA 397 which targets the lackase messenger RNA. So this is already known, the 397. When we search in the Arabidopsis, we found there are two 397, 397A and 397B, and there is only one nucleotide difference. Okay? And we found that on 397A does not respond to any kind of stress. We tried with the different stress. This is mannitol. 397A does not. It remains the same expression. But 397B is highly expressed in the mannitol treatment, in any other treatments also. And this 397B also expresses in the where the lignin deposition occurs, that means in the vasculature. So that means they express in the same domain and the same tissues. And when we express this microRNA in the plant, we found this LAC2, the level goes down the most. So this is the major target of the microRNA 397B. Lackase 4 also that goes down, but lackase 17, because LAC2, 4 and 17, these three are targets bioinformatically. You can say that they are the targets of this 397. But when we did in the assay, we found it does not target LAC17 in the system. It targets LAC4, but it targets mostly the LAC2. Okay. So LAC2 is the major target of microRNA 397B in the Arabidopsis root. Now we did number of experiments. Yesterday Ashish also explained what is STTM, and that if you want to knock out or not knock down or inactivate the microRNA, this is the best way to do, or the easier way to do, because you can just design, synthesize the target sequence, and you make it the double target sequence, and with a spacer, and you change this nucleotide in the cleavage site, so that the microRNA will bind there, but cannot cleave this one. So it will absorb like a sponge. So if the microRNA is absorbed like a sponge, so microRNA will spare its actual target, the native, the cDNA native target. So by that way you can inactivate the microRNAs. So this is the LAC2 knockout I showed you before. Whenever you overexpress the microRNA, that will show the similar phenotype like LAC2, because if you overexpress microRNA, then it will degrade the LAC2 and it will remain the same. STTM, when you inactivate the microRNA, then its route is longer, and we all in a LAC2 knockout, we expressed a LAC2 cDNA which cannot be cleaved by the microRNA 397, because we have changed this cleavage site without changing the amino acid. Because you know the codon, there are a number of codons can be used for the one amino acid. So we change the codon so that amino acid remains same, but it cannot be cleaved by, it is resistant to the cleavage. So they are also showing the similar or longer phenotype than the Columbia, which is wild type. So this is the root length, and this is the lignin estimation as well as the lignin staining. So in LAC2 I said that is constitutively showing the high lignin, same the microRNA overexpressing line, but in the STTM, in the resistant LAC2, it shows the less lignin deposition, and that's why the roots are longer, because if lignin is deposited, root growth stops. The same thing happens in case of the phosphate deprivation. So if you remove the phosphate, this is the normal phosphate concentration in the synthetic media. If you remove the phosphate, again this LAC4 and LAC2 expression goes down, and the microRNA 397B expression goes up. And the same thing happens here if it is a low phosphate. And in case of mannitol, we saw the lignin deposition in the elongation zone of the root. Root has a different zone, meristematic zone, elongation zone, transition zone. But now the lignin deposition is close to the meristematic zone. Okay. And this is the all different kind of experiment. You can see if there is a more lignin deposition, there is a more iron deposition also, because as it is aromatic, iron cannot go inside the vasculature, so they remain stored in the apoplastic region. And if the iron is there, we have measured there is a huge ROS generation, and because of this ROS generation, you can see more calcium, callose deposition. If there is callose deposition, then root growth stops, and it is irreversible. So another lateral root takes part as a tap root, as the major root, because this root growth totally stops. And we have shown that this is also dependent on whether you are overexpressing microRNA or not, or you are inactivating the microRNA or not. So the microRNA has a role to play because the microRNA 397B goes up, LAC2 goes down, and as LAC2 is the negative regulator of the lignin deposition, if LAC2 goes down, there is more lignin deposition, root growth stops, and the plant become more drought tolerant. That we are not showing here. There is another slide. So this is the Arabidopsis story. Now in the chickpea, we wanted to apply the same thing in chickpea because chickpea is a crop, nobody eats Arabidopsis. So we have to show it in the chickpea so that we can translate this result in a crop plant. So in the crop plant, we grow it like a crop. We have a big pot and with the soil full of soil, and as the drought comes, we stop irrigation, right? The difference between the synthetic media and the soil is when you apply the mannitol, it is evenly spread in the synthetic media. The whole root is experiencing the same concentration of the mannitol. But if you stop irrigation in the soil, the top surface will first dry, but there is still moisture in the bottom surface. So there is a gradient of soil moisture concentration there. So as the top surface is dries and the bottom surface still has the water, we have seen the root elongates. In mannitol, it stops because bottom there is also mannitol, top there is also mannitol, so everywhere is mannitol. But here in the soil, it is in the gradient, so it's, but we have seen the cell number does not increase, the cell elongates. In the meristem, the elongation zone goes down, it very close to the meristem, it starts the elongating the cell. Cell number does not increase, not the cell cycle. But the same thing happens in the elongation zone, we can see the more lignin deposition. Okay. So we have estimated the lignin, we have stained it with the lignin. So chickpea has the 20 lackases, and we found that there are many, 10 of them show the high expression and none of them show the down regulation like the LAC2 and LAC4. All of them are showing high expression. Okay, out of 20, 10 are. But, and this chickpea microRNA 397, in chickpea there is only one microRNA 397, no A or B, and its sequence matches with the 397A of Arabidopsis. That goes down instead of going up in Arabidopsis, in chickpea it goes down in a normal field situation. Okay. So this is all the microRNA, it is very conserved, and only Arabidopsis has this 397B whose this sequence is different. Otherwise, rest of the crop plant, they have the same 397 sequence, and they have only, even if there is the 397B in soybean, it's still the sequence matches with the 397A. The Arabidopsis is the exception here. And bioinformatically it targets LAC4 like LAC4L or LAC17L, but when we expressed in the plant, we found it targets only the 17L and 4, not the LAC4L. So it does not target the LAC2 at all. So this is the difference between the Arabidopsis and chickpea. And we did this STTM overexpression, whatever we did in Arabidopsis, we repeated this thing in the chickpea also. There in the STTM, you can see exactly the opposite, because now there is no LAC2 story of LAC2, there is no negative regulator of the lignin deposition. Everybody is the positive regulator of the lignin deposition, and this microRNA goes down and the LAC goes up. The same thing we have when we stained with the lignin, we found that in the drought it increases, but in microRNA overexpression line you can see the less lignin deposition because it degrades the LAC4 and LAC17L, and STTM there is more in the drought, that is there. And as I was showing, as I was saying, that there is a lignin deposition in the xylem cell wall. So we measured in electron microscope, we measured the cell wall thickness, and it is really that whenever you are overexpressing the microRNA 397 in chickpea, the cell wall thickness goes down, and that's why the radius becomes more, and the STTM, the thickness is more, and that's why the radius is less. And this is the lignin deposition, and there is a different parameter of drought tolerance, and we have seen if you have more lignin deposition in STTM, it becomes more drought tolerant. Now the chickpea has a very devastating disease which is called the dry root rot, which is infected with the fungus Micropomena fasciolina, and it is a really problem in the Marathwada region, because once it comes, the next morning you can see the whole field becomes a yellow. So when I was in the research advisory committee of one of my colleagues, Dr. Senthil Kumar, and I saw that whenever this Macrophomina fasciolina infects the chickpea root, there is a lignin deposition, and there is a the infection varies with the lignin content of the root. So then, I took the fungus from him, and then we found that this is, and we applied in all this transgenic line. Now we have, one story I should say, that apart from the vasculature, lignin is also deposited in the exodermis. What is exodermis? Just below the epidermis. Okay, the next layer of the epidermis is the exodermis. And chickpea is a very rare legume. Some of the legume have the constitutive lignified exodermis. Generally plants do not have the lignified exodermis. If you treat them with any stress, then only they deposit lignin in the exodermis. But the chickpea is one of the rare, like chickpea and what is the cashery? That is the lathite. They have all these drought tolerant, this legume, they have constitutive lignin deposition in the exodermis, and that's why they are drought tolerant. So I have one collaborator in Netherlands. She has tested 25 legumes and found that only chickpea and lathyrus, they have constitutive this lignin deposition in the exodermis. So I tested in those and found that there is a constitutive, this is only the stress condition. I have the picture of the normal condition also. The infection, this is infected conditions. So this is the exodermis, and whenever you see the messenger RNA overexpression, microRNA overexpression, and there is less lignin deposition, the fungus can invade the exodermis by degrading the cell wall. You can see the degradation of the cell wall, and this is the lignin. And when the STTM is there, that means there is more lignin deposition. It is a continuous cell wall. That means the fungus cannot go inside. So this is the stain of the fungus staining. It can go and infect more layer within the cortex, and we have assayed the fungal DNA content also. So this is the STTM. It is almost fungus cannot infect this STTM chickpea lines, where this is the microRNA overexpressing line, this is the control, and there are other assays, how many layers are there. So we started with the abiotic stress but ended with the biotic stress, and found that microRNA controls the lignin deposition in the root, not only in the Arabidopsis and chickpea, but the mechanism is different. So there is the microRNA is going up, there is microRNA goes down, there is a lacase 2 which goes down, and here all the lacases 4 and 17 that goes up. The story is just opposite, but at least one thing is common. Okay, there also 397 targets the lacase, here also 397 targets the lacase. That's the message or lesson we can get it from the Arabidopsis. But your crop as a species and as a growing system is different, so you have to work on the crop individually. You cannot directly translate your result from the model plant to the crop. So that is the bottom line message I wanted to give. And so these are the summarized what I said. This is I summarized here. This is the difference, and this is the alloc already described. This is our institute. So that's it. And any question? If yes.
This STTM in chickpea, there is a lignin deposition in exodermis, but there is no change in xylems.
Yes, there is xylem.
Okay, but actually I don't know. Maybe I missed it. I thought it's only in the exodermis.
No. In there, I have shown the exodermis, and that slide only.
Okay.
And I took only that part. I didn't go to the xylem. The picture is only on the.
So will it protect the other pathogens, which are xylem growing pathogens?
I have not tested because I am not a biotic stress person, you know. So I just got the idea from there and did only one experiment. Somebody works on chickpeas, so she might know. Through the xylem, I didn't work with the fusarium.
Correct me if I am wrong. Okay, so your this MIR 3397 goes down in case of chickpea. It goes up in Arabidopsis.
Right.
And it acts during, this is during drought stress, right?
There is a mannitol. Mannitol you can say here.
Chickpea also drought.
Chickpea drought, really drought, the field drought.
So when it is going down, how do you think this is going to regulate this lacase when the amount of MIR is already down?
Yes, but in normal condition, you will see the lacase level is down. See, in normal condition it controls. So whenever in drought it goes down, so it cannot control obviously the normal goes up.
Normal.
So
This drought, what you observed in laboratories, cannot extrapolate to chickpea.
right, because the system is different, na? Because there is the mannitol everywhere.
One more question.
Yeah.
How did you, out of this, all 20 lacquers in chickpea, right? All getting upregulated?
No, 10.
10. So how did you confirm that out of 10 only this one is getting target of 397?
But it is bioinformatics.
It may target some other.
No, we have tested all the 20 and none was is going down. Yes.
Why you are not considering suberin?
Yes, that's because the suberin, there is a paper in cell, Nicogel. So laccase does not affect the suberin.
Okay.
So my concentration was on laccase only.
Actually, we are working on the same thing in the rice, cascade
Right.
strip under the salt tolerance.
Right.
So there is also one mRNA involved. That is,
Right.
we are studying only for suberin deposition, not the lignin.
But laccase is not involved in the suberin. That paper is there.
Okay. Thank you.
Yes. No. I do not work with fungus. No. If it grows through the xylem, I don't or phloem, then I do not know. No, I did not check the yield, but in normal condition, I did not see any difference in yield. But under the drought, whether there is any change in yield or not, I did not see that. Obviously, it will compromise the yield, yes. But one thing I am telling you, you know Vincent Valdez of this ICRISAT, he recently went back. He was showing there, he has given a very nice review. So this drought tolerant rice variety Dular has the smaller, narrower xylem diameter and more lignin deposition than the drought susceptible IR64. So he has given a number of rice varieties name where the lignin deposition is more and the xylem radius is narrower, and they are equally yielding, high yielding. I don't know, IR64 is the highest yielding. Obviously, you cannot compare the Dular with the IR64, but they are regularly yielding. So, okay, the Dular is you know better than.
Dular is internationally self incompatibility donor gene.
Okay.
When Chinese are using the hybrid variety, using.
So I don't know. So normally it does not affect the yield. Medicago, there is only one microRNA, and I imagine there should not be any difference between the Medicago and chickpea, because whatever we have seen in Medicago, that easily works in chickpea, because these are all the galligoid clade of the legume, and they behave same. Milletoid, they behave differently, like soybean.
Okay, thank you. If
Okay.
there is no other question from the student.
I was expecting, anyway, because that's what I kept talk very simple, not very
So
complicated.
if there is no question, then give a good applaud to you.
We'll take a 15 minutes tea break and gather back after 15 minutes for the next session, so for the next set of lectures.
Can I just see once?